NER1 is a transcription factor that was discovered about 15 years ago as a key regulator of dopamine neuron survival in the brain. In development, it is essential for survival. If NER1 is deleted, then dopamine neurons will not will die. Interestingly, NER1 is also expressed in the adult brain and plays a role there as a regulator of dopamine neuron expression, that is, genes that are involved in dopamine neuron transmission. Interestingly, NER1 has been linked also to Parkinson's disease. There is a number of publications, you see 4 examples here, that have identified mutations or polymorphisms in the NER1 gene linked to Parkinson's disease. It doesn't mean that NER1 is a Parkinson's disease-inducing gene, but it may be a susceptibility factor. Jeffrey Cordover in Chicago has a few years ago made a study in Parkinsonian brains showing that NER1 is indeed downregulated in patients with Parkinson's disease. And you can see here on the slide that the level of NER1 in dopamine neurons is perhaps only half of that seen in age-matched controls. And the level of NER1, in addition, is correlated with the level of tyrosine hydroxylase in these neurons. So in disease-affected neurons in the Parkinsonian brain, NER1 is indeed downregulated. We have been able to reproduce this situation in rodents, in rats and mice, by overexpressing the disease-causing protein alpha-synuclein in dopamine neurons. And you can see on this slide that in animals where alpha-synuclein has been overexpressed using a virus vector, an adeno-associated virus vector, NER1 is reduced by 60-70% in the dopamine neurons. And this is accompanied by a reduction in both tyrosine hydroxylase, TH, as you see here, as well as the GDNF receptor, RET, which is a very interesting observation. This suggests that GDNF signaling may be affected in neurons that are overexpressing alpha-synuclein. So, as you see in this slide here, we've shown that GDNF receptor RET is downregulated in alpha-synuclein overexpressing dopamine neurons. And indeed, GDNF fails to protect against alpha-synuclein toxicity in dopamine neurons, which gives or raises the possibility that NER1 is involved in regulation of the dopamine neuron's ability to respond to GDNF through downregulation of the receptor. We've done an experiment illustrated here where we overexpress alpha-synuclein first. We wait 12 days when the level of alpha-synuclein is increased. And at that point, we give a bolus injection of GDNF, 1 microgram, into the striatum or over the nigra, and look for the trophic response in dopamine neurons. And as you can see here, in this Western blot analysis, the activation of downstream effectors of RET signaling, the phosphoric, phospho-AKT, phospho-S6, and phospho-BAD, all these downstream molecules are not responding to GDNF in cells that overexpress alpha-synuclein. And as a comparison, we have also made studies in a 6-hydroxydopamine toxin model where the response is perfectly normal. So it seems that the triggering of the response by activation of RET is blocked in neurons affected by alpha-synuclein. We've also shown that deletion of Neur1 as itself, the transcription factor, is producing a similar effect. So if we remove Neur1 in knockout mice, we can see that the response here illustrated by phospho-AKT and phospho-S6 is significantly reduced in dopamine neurons. And to the left you can see phospho-S6 immunostaining, which again shows that in Neur1 knockout mice, the response monitored by the phospho-S6 upregulation is indeed blocked. And conversely, we've been able to show also that in alpha-synuclein overexpressing cells where the GDNF signaling is blocked, we can restore signaling by putting back Neur1. So in this experiment, we combine alpha-synuclein overexpression with overexpression of Neur1. And in this situation, the signaling response from an injection of GDNF is restored to normal level. So in conclusion, I think we've good reasons to believe that alpha-synuclein is impairing the dopamine neuron's ability to respond to trophic factors, in this case GDNF. And this is mediated through downregulation of the Neur1 transcription factor. We think also there is good evidence to suggest that alpha-synuclein toxicity and the failure of GDNF to protect against alpha-synuclein toxicity is due to reduction of RET expression as a consequence of the effect on Neur1. Neur1 conversely is effective in restoring RET expression. It brings back the receptor to the dopamine neurons, and it makes it possible again for GDNF to induce signaling. All these data, they point to Neur1 as an interesting new potential drug target. Neur1 is a nuclear receptor, orphan nuclear receptor, which is among the druggable kind of nuclear receptors, which means that biotech companies and drug companies are currently involved in developing Neur1 activating drugs or Neur1 agonists that can be used to boost Neur1 expression or Neur1 function in dopamine neurons. This may by itself be neuroprotective, protecting the dopamine neurons against alpha-synuclein toxicity. But interestingly, such agonists or activating drugs may also be used to potentiate the effect of GDNF as a neurotrophic factor, as a survival factor in dopamine neurons. And this may be a very interesting application or use of Neur1 activating drugs in the clinic. Thank you.